a chance to sit through Dom's session that just finished up a few minutes ago? And if not, at the very least, welcome for the first time to our session tonight, where we're going to be actively reviewing the diagnostic test that you took and taking a look specifically at the bio and biochem portions. Now again, these are absolutely opportunity areas for you to go back and look over the test that you just took and get a better idea of what to be looking for when you're actively reviewing that test, as opposed to maybe just going back down through and feeling a little bit lost or confused, or not necessarily knowing which questions are going to be the most benefit for you to take a look at. As we jump in, what we're going to be covering are a few main points. First of all, I want to make sure that you understand what the MCAT is actually going through and trying to test you on in bio and biochem. Take a look at the reasoning and strategies we can be using, and then figure out a little bit more about how to be interpreting passage material and get the most out of a passage in the first place. Now, to be clear, what we're trying to do is actively review the diagnostic you just took, so all of the questions that we're going to be using are based off of your diagnostic test. Additionally, though, when you go back and review, you should be using the same sort of analyses that I'm going to be showing you in today's session. Let's jump right in. First of all, the MCAT actively tests in bio and biochem. Well, this is going to be a shocker. Bio and biochem content. So questions like this are actively foundation level bio and biochem material that we absolutely have to know going into the exam. That said, this is a question that you would see in an introductory biological course in general, where you'd have to go through and memorize the different dermal layers, and then this would be a fast point. But help me out down here in chat. Even if you didn't remember what dermal layer the glomerulus came from, what could you get rid of? This is a pure content question, for sure. We can still do some critical thinking. Yeah, notochord, thank you very much. Unless we're talking about a specific spinal development, we're not talking about that. We're talking about a dermal layer. Nicely done, James, Maribel, Ariel, nicely done. So again, there is some content memorization, and the MCAT expects you to know the 3 dermal layers and what the notochord is, but we could go one step further. Help me out. You know where the glomerulus should be from like a Bio 101 class. An undergrad should be kidney, absolutely. Now the kidney is doing something specific. The kidney, and notice the way I'm talking through this problem, this is the way you would hit a problem like this. Even though it's a memorization content, we can still go through the systems. Okay, kidney is moving something actively through some sort of process. The kidneys are part of a movement process. Well then, movement process is mesoderm. And this is the sort of thing that we talk about in some of the review videos. If you've already glanced through the kidney review on your online syllabus and looked at some of those extra science and demand, but the whole point is, we can still work through content questions. We do need a foundation level of material to answer it in the end, but this is a sort of level that the MCAT expects you to know for some of this in terms of embryology in this case. Now certainly we would have to know things like blastula, gastrula, talk about the different dermal layers and development, and all of this, again, is part of your foundation content, but this is the sort of thing the MCAT could ask. They could also ask a question that is very specific, that is dealing in this case with genetics. Well, again, we'll talk a lot more about genetics in the overall course, but this is a question that a lot of people see on their diagnostic test and then don't necessarily remember which exact one is referenced by this idea of starting at a large number, going to a small number, going back up to a large number. Now, even if you had to guess on this one, I want to come back to that kind of critical thinking for a second. This is content we have to know, but could we get rid of at least one? Is there at least one in here we could get rid of, because it's clearly not dealing with just a number. Hardy-Weinberg equation, Maribel, Catherine, is the first one I would get rid of personally as well. Because we do not have any allele distributions, we can't be using P's or Q's. So definitely nothing about the actual allelic distribution. Gone. Now I see Suhag is saying maybe we could also get rid of C. Any takers to back up getting rid of C? I want to make sure we're doing this as a team. This is the sort of thing that, unfortunately, you don't get to do the MCAT as a team, but I want to see that any others we can probably get rid of. We've got founder effect, allopathic, and genetic bottleneck still left. What's one more we could get rid of? Okay, Maribel says get rid of founder effect. Now again, we've got a vote for B and a vote for C to eliminate. I need some more takers. We're a team here. I just happen to be on camera. You're working with me. Okay, a second for founder effect. Okay, now founder effect is something that we could see a small population and a large population boom afterwards, but specifically founder effect is referencing the variation based off of a population difference. Again, this is something we talk about with people who have polydactyly, or the six-finger effect, or the prevalence of a particular hair color, eye color in certain regions of the world based off of a particular subset variation that is prevalent that then becomes an overall variation within that particular population. So red hair, for example, or six fingers would be a founder effect. Now, at this point, we're left with a 50-50 shot. Something about allopathic speciation or something about genetic bottleneck. If you get down to 50-50 on test day, doing this sort of elimination, excellent. Do not feel bad. Remember, on the MCAT, I love to say this to all of my students, you need to know 3/4 of the material. Why do I say that? Why don't I say 100%? Well, because if you know 3/4 of the material, either you've picked out 3 answers that you know are wrong, and the last one is the right one, or within the 3 things that you know out of 4, you've gotten the right answer. So in this particular question, noticing one word in C helps us get rid of it. And the word is not allopathic, which is what most people look at first. It's speciation. Have we created new species in the new subset of individuals? Oh, it didn't say anything about us actually doing that, huh? Well, then we can get rid of it. No new species have been formed. It must be answer choice D, genetic bottleneck. And yes, genetic bottleneck by definition is a change in the overall demographic total that goes down to a small number and back out to a large number. Think going through the neck of a bottle is a small, and then coming back out of that neck, we can flow out to a larger value. So exactly right. Wolf population, Suhag, is another one. Wolf population went to a distinct bottleneck and is now expanding back out again. A lot of other species we could talk about that do bottlenecking, unfortunately usually due to human involvement. But yes, another good example. Now notice, what we're talking about here are content-specific information, and we want to go a little beyond that. Because I agree with you, Suhag, and I can give you book explanations and book references for a lot of these different things, but the MCAT isn't necessarily going to pick that. So do be on the lookout for different ways to express some of the biological content and some of the biochem material. For example, off the top of my head, the biochem content that I love to test is specifically the different forms of amino acids. You have to memorize all of those by testing. So on test day, I want to have all of those memorized. I want to have all of them written down, but they could still give me a brand new molecule that maybe is related to amino acids. Maybe something like methotrexate, right? Methotrexate has carboxylate groups, has amine groups, has a bunch of different subspecies that look very familiar for a biochem and organic chemistry material, but you maybe have never ever seen methotrexate at all. So again, coming back to what do I notice in the concept? What of the structure references back to stuff I have memorized? That's a key piece. Yeah, absolutely, Sivak. As many variations as possible. Because even though the MCAT has to test you on particular content subcategories, because it's a standardized test, they have to, they can give you different examples of it each time. And that's a great opportunity for us. It's a little bit of a sidetrack here, but let me give you another example that I love about that. And this is again a little bit of biochem. It's almost into the gen chem category. Here's my question. I know it's not necessarily what the slide is on right now, but let's go a little sidetrack because Suhag gave a great example here. Suppose I tell you that I have this molecule that forms in an aqueous solution. And I want to know, this is absolutely the type of question that they could feed you on the MCAT on test day. And I want to know, this molecule has formed in an acidic aqueous solution. What type of bonding is it an example of? I love this question. It is my favorite new question. What type of bond? And there are a lot of types of bonding. I know you're at the beginning of your Kaplan course. Maybe you haven't had a long chance to look at ionic bonding, polar bonding, hydrogen bonding, dipole-dipole interactions, covalent bonding. All of these are different types of bonding you could see that fall into the subcategories, exactly, Emily, of inter versus intramolecular bonding. Now, the reason I ask this question is exactly because of the conversation I'm having with you all. Ooh, Suhag is real close. Real close. I love this. Because this is an example that I really like because it's not the textbook example. If I had given you this example instead, what type of bonding am I talking about here? Yeah, Maribel, 100%. And when I ask this question, I get the right answer. Look at that, within like 7 seconds. This is the textbook example. Ammonia bonding with boron trifluoride. Right? We have an extra lone pair here on boron. That extra lone pair gets donated over to boron. Then boron has a perfect octet. It's happy. And nitrogen retains its original octet. It's happy. But that is exactly what's happening in water. Here is our oxygen with a couple lone pairs left over. And here is our hydrogen. Here is our hydrogen that's floating along on the outskirts with nothing. So the lone pair from oxygen gets donated over to hydrogen. Hydrogen is now happy, full S subshell, and oxygen still retains its octet. This is coordinate covalent bonding. See what I'm getting at here? I'm getting back to Suhag's question earlier about the difference between knowing the textbook example and knowing the concept behind it. That we have something that is now being absorbed. We're absorbing some new atom in this case, or some new molecule in the boron trifluoride example, due to there being a lone pair available for bonding. So this is absolutely lone pair bonding geometry, coordinate covalent bonds. Cool, good work. Now again, the takeaway from this, as with everything on the MCAT, is to understand the concept behind it, as opposed to just memorizing 1 or 2 facts, or in the case of what we were discussing a second ago, memorizing 1 or 2 examples. We should always know the methodology or the process or the system, because that's what the MCAT loves to test consistently. Now, speaking of which, problem number 28 is a great one. I've shown you the first 2 lines of it, but I haven't shown you the 3rd line, and this is for a reason. The way they have designed this question, where they're talking about the liver being responsible for changing alcohol to some other products, the way they designed it is to give you this at the very beginning of the question and to get us a little sidetracked. That's not the main point of the liver. Right? If it was the main point of the liver, then we would assume that a standard diet in every single human is alcohol, and that we have to have this alcohol processed, otherwise we would die. Now, some parts of that could be true for certain individuals, but it is certainly not the main function of the liver. In fact, it's not even the main concern for the question. The question is actually asking about what is not a function of the liver. And this is the sort of thing that on the MCAT, when you see a question that has a long lead up or that's based off of a passage that has a lot of complicated material, always give yourself that time to do the assess and plan of a Kaplan strategy. What is really being asked, that's our assess piece, and then plan, how can I go through and answer this? Well, a simple way to go through and answer this would be elimination. Which are ones that the liver does do? Well, the liver had better do something with bile, so get rid of answer choice D. The liver certainly deals with gluconeogenesis, absolutely, and in order to have proper functionality, it had better be making some sort of cholesterols. So we can get rid of those answer choices that are true functions of the liver and be left with urea excretion. Its primary process is not for urea excretion. Bonus question. The liver is involved in urea production. It's the site of the urea cycle, but who or what system is involved in urea excretion? Overall excretory system, absolutely right. Talking about renal system and kidneys overall, definitely. Catherine, Emily, Ariel, I love this. Because look at what you're doing. You're going one step beyond just the pure content to attack the MCAT as a systematic test. That's the beauty of this. We're going through right now, all the content I've just covered, this is basic biological content that the MCAT is definitely going to ask you about. Renal system, excretory system, talking about cell biology or embryology. This is content that over the next several weeks you are going to get flashcards, review notes, watch videos, and get this incorporated. But then, one step beyond that is always this sort of critical analysis and thinking. Let's do one more of these where, again, you might never have seen methotrexate. This was the question that I pulled up a second ago. But it's telling us a little bit about it. So it's really asking what is not likely to be seen in someone suffering from parasympathetic damage. Well, even if they had not given us that hint, they're asking us which is least likely to be seen. Well then, in that scenario, and in any scenario, when they say most likely, least likely, which of the following is not, except, all those variations, what they're really asking you is, can you recognize? Write this down in your notebook if you haven't already done so. What they're really asking you is, can you recognize that one is different and three are the same? So let's run down the line. Tell me this is a sympathetic response, like a fight or flight, or this is a parasympathetic response, like rest and digest. A, pupil dilation. Give me an S or a P. S or P. See a lot of S's here. S, 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 sympathetic. Absolutely. Pupil dilation, getting large, getting in more light. Okay, more processing. Great. How about the next one? Stopping motility of the bowel. S or P. S or P, S or P. Stopping motility better be sympathetic as well. Okay, now at this point, pause, because I love the answers I've been getting here. This is awesome. Could A and B both be right? No, right? That was a softball question. I know they can't both be right. Bonus question. Could one of them be right? It's still no. So James, Rachel, Maribel, you got what I was getting at here. It's still no. First of all, they can't both be right? They never on the MCAT can two answer choices both be right. Second of all, we're being asked for which one is different, which one is least, which one is the except. So A and B are both sympathetic responses? Well then, if they can't both be right, then they must both be wrong. In fact, as soon as we get to C, more blood supply, more vasodilation, we know the answer must be D. It's the one that is not a sympathetic response. And in fact, decreased breath rate is like rest, digest, sleeping. That is parasympathetic, and it must be the right answer. See what I'm getting at here? This is consistent for all of the lessons that I personally teach, as I always want you to go one step beyond the question at hand. Sure, question 13 is a great question. It's a very classic indicative question the MCAT loves to give you, the least likely, the least similar, the except question. But the way we attack it is going to be consistent no matter whether it happens to be about parasympathetic or it happens to be about excretory system or respiration. That's the takeaway from this one. Two answer choices can't both be right. And if they're about the same concept, like sympathetic, then they must both be wrong. Now, a nice segue from this is a question like number 29, where we do have to know content to answer it, and they've also given us some hints anytime the content could otherwise overlap. So we clearly see that we have little spherical balls here that are arranged in chains. Now, since they're little spherical balls in chains, we can probably get rid of one or two of these. Which two specifically, before we get into the next hint? I mean, at least one we could get rid of. We can get rid of A. Certainly it's not a bacillus, correct? And C is the other one, Rachel, Ariel, that I usually get rid of. Is a spirals. We are not seeing that form either. So, between the remaining two, it certainly could be a virus. That could be a really small virus or some sort of virus arrangement that we're just not familiar with, except it tells us it can't pass through a 0.1 micrometer filter. Now, it can't pass through is really telling us it's not super small. It's big enough that it would get blocked. So we think, okay, bigger than 0.1 microns means it can't be a virus. The only thing we're left with is a coccus. Think, classic example of pathogens that are spherical arranged in chains. Well, classic example to the question that we always get. What would be a classic example of a coccus? Sure. I see Streptococcus would be a classic example I would normally see. I see Suhag also with a couple more. Again, one of the great things to do on test day, if you're ever presented with a new term or something that you're not familiar with, like a bacillus or spirals, if you haven't seen those terms before, or when you were taught them, they were just like a vague concept, lock it down with something. For example, for a coccus, I always think Streptococcus every single time. It's not that small, it's circling chains, and I've got that immediate connection to a real-world example. So I know this sounds like I'm going counter to what I told Suhag a second ago. The point that I was making earlier is that don't just know the one example, know the concept behind it. The point I'm making right now is that if you have an example that helps you understand or helps you memorize or helps you flashcard, absolutely use that, because that's going to help you get to that connection point. Now again, we had to use a little critical thinking here, but a lot of this question was memorization. What we're kind of showing to you in this first section where we're running down through all of the different content that the MCAT might bring up is to remind you that there is a wide, disparate variety of questions they could ask. So the first at least half to 2/3 of your course, you should be spending a lot of time going through the science videos, the review notes, doing flashcards to try and get at least the foundation down first. One of the things that I do for Kaplan besides doing these sessions is I'm actually part of our learning science team. What we've found is that getting a good foundation early and then in the last half or third or so of the course repeatedly testing yourself is the best way to make sure that content stays in long-term memory, and also to make sure that you have that connection to learning the content and then seeing how the MCAT actually tests it. Now that said, we do also get a lot of reasoning questions on the MCAT where you might be presented with brand new information or information from a passage or information you had to memorize in theory, but maybe you missed it. Or maybe you're taking full-length number 1 or full-length number 2 and you just haven't gone through all of the material yet. So you hit a question that has content you haven't seen before. Well, first of all, a question like number 5, we wouldn't really be able to answer without knowing what creatine clearance means or what it relates to. But I love to do this with all of my classes, and you're my students right now, so we get to play this game. I don't know the answer. I don't remember creatine clearance. Maybe it has something to do with liver or something to do with metabolites. I'm not sure. But I've probably seen creatinine before. Have you ever seen that word before or seen it in reference somewhere before? Okay, 1 or 2 people have. Where? We're in the real world. Let's think real world here for a second. In muscle building, usually. Exactly, Emily. In the gym, Suhag? Okay. Now, those of you saying blood tests, those of you saying kidneys, you are right as well. Right? Classically, clinically, you are absolutely right. That is where we would normally see it. Kidney filtration, Ellen? Absolutely. Absolutely. But what I'm getting at here is suppose you didn't know what the actual functionality was. You have a connection to things like the gym, bodybuilding, protein shakes. Okay, so those people who are taking extra protein shakes, doing supplements, going to the gym, are they having a really high fat diet? Okay, let's get rid of B then. Are they having a diet really high in a lot of sugars? Hopefully not exactly, James. So A and B are gone. Now, between C and D, you might be tempted, right? These 2 answer choices sound reasonable. A person who's working out a lot, who's going to the gym a lot, probably isn't eating a lot of saturated fats, so C sounds tempting. But the connection you've already made, protein shakes, supplements, matches up perfectly with answer choice D. So the right answer for number 5 is D. A diet high in proteins would have a high creatine clearance. Now this is a fact you could memorize. This is a fact that when you go back to kidney synthesis, when you talk about filtration, you will see creatine clearance referenced. It is something that we do cover throughout the course, but it's something where we can use our common sense to attack it. Let me contrast this, though, with question number 9. Do you have a lot of real-world scenarios where you've connected creatine with ADH or creatine with vasopressin? Probably not this one, right? So part of being able to answer questions efficiently, being able to critically think through the ones that you can actually handle, is also doing what we cover a lot in the Kaplan course, triaging. So question like number 9, if there is no real-world example, if there's no answer choice that can quickly, easily be eliminated, to do some triage. Question number 9, let's skip it. Maybe we'll mark it. At the very least, before the test is over, you're going to guess on it, because remember, there is absolutely no detriment to guessing on an answer choice. So at the very least, we'd guess, but we'd move on to another one, well, maybe like number 12. Now at this point, I want to let you try and answer number 12. Down here in chat, methotrexate does some maximal cytotoxic effect in one or many of these different phases. So help me out down here. Let's think about this as a team. What are some clues in the question stem? Because we can't answer this without at least some knowledge of either methotrexate or of the different phases. What are some clues? Okay, cytotoxic. That sounds pretty bad, Suhag. Sounds nasty, Nicole. So cytotoxic sounds like it'd be a really bad thing for us to have. Cyto sounds like cytokinesis, right, which is cutting. So maybe that sounds like maybe it would be part of some sort of cutting phase or some sort of disruption phase. But it's toxic to some sort of thing. So toxic to cells, exactly, Lon. And there we go. That's the real root, is kills. Exactly, Nicole. So it's killing something. It's toxic to something. James is doing the exact critical thinking I want to see on this one. And we're talking about cell cycles. So where would killing something do the most damage to replication? Definitely in 2, because 2 is S, right? S is stuff phase. S is DNA replication, right? If we cannot make more DNA because we have this methotrexate, then we can't get enough stuff to even get to G2. So this is a great MCAT style question. I love this one because it's really classically indicative of the sort of almost mean traps that they throw at you. Notice the way they've asked you this question. Where does it achieve its maximal effect? Well, hold on. Let me give you one piece of information. All right, and then we can talk about whether C is still the right answer. Methotrexate, which is also called a methopterin, is usually a cancer-fighting drug. And what it does is it specifically targets DNA. And it makes it so that, that thymine can't be produced. Okay? So where is that going to affect the most? S phase. Now I know what you're thinking to yourself. You're thinking, hold the phone, Eli. Well, wait a second. Won't that also affect M phase? Right? Because I'm completely shutting off M phase. If I can't build enough DNA, I'm never even going to get to M phase. But that's the point, Maribel. You absolutely hit the nail on the head. Because I'm essentially killing S, I can't get to M phase. So maximal cytotoxic effect, boom, absolutely answer choice B. It is destroying S phase. We cannot make new DNA. Well then, we're not affecting the M phase because we can't even get there. Now again, this is a sort of thing where I like what James put it here in chat because that's what I thought when I saw this problem. I thought, it sounds a little aggressive, sounds mean, sounds tricky, but it's not actually, right? If they're asking for effect, they're asking for what it stops, what it's impacting. It's like, here's a good example that I like. There was a teacher who was going to come to class, and she had to do a couple tasks before she came to the classroom. First thing she had to do was get in her car, drive to the classroom. Second thing was she had to change into a new shirt, new blouse before coming into the classroom. And the third thing she had to do was teach the lesson. Now her car broke down. What was affected? She had 3 tasks. Must drive to class, must change blouses, must teach. The thing that was affected was driving. She can no longer complete driving. The other ones have not been directly affected. They have been indirectly affected. That's what we're getting at here for number 12. And that's a question that I have seen repeatedly on the MCAT, the old versions and new versions, because this is the sort of thing that if you overthink it and think, well, these other ones are completely stopped, we can't even get there, you're missing the point. The point is what's being affected, what is being stopped, what is being actively destroyed, or in this case, killed, right, cytotoxic. So the right answer has to be B, and we can work through it together with that kind of critical thinking. Now, the flip side to critical thinking, to reasoning, is being able to go through a larger passage and larger material in order to actually reason through what's going to be tested in the first place. So when you're critically reading on the MCAT, and we go through this a lot more in the Kaplan course, what we're really trying to do is analyze what's going to be tested, so that when you hit the question set, you're already relatively prepared to answer those questions. So what I'm going to do is actually spend a decent amount of time here talking about how to go through a passage, what to look for, and what to actively expect in the question set. And then we'll maybe have time to let you walk through and do another passage at the end, or at the very least, to go back down through and when you're reviewing the rest of your bio and biochem content, do your diagnostic review of other passages that you thought were hard and do those for homework. So the idea here, before we jump in and actually do it, is to try and pick out what's most testable, what's actually going to come up in a question set. So when I'm reading down through this paragraph here, paragraph number 1, I could write a quick outline of it, like this. Something about homocystinuria background and some inherited forms of genetics in here. But what I'm actually going to be asking myself while reading is what's likely to be tested. Now I'm going to put that back to you. You've already done this passage. You've already done this. What within paragraph 1 either was tested or was important to know to answer the questions? Gene impact, absolutely, Suhag, and that it was an inherited genetics. Now it gives us a bunch of hints. And this is what I'm going to turn you on to at this point, is the MCAT loves to test certain content more than others. So if you notice, we've got inherited disorder. So it's some sort of overarching disorder that we're going to be seeing consistently on this. And additionally, we've got scanning down through some sort of primary gene. So it's a gene mutation, exactly right, Maribel. And then it's an autosomal recessive, so maybe I'm going to get a question about autosomal recessive versus autosomal dominant versus sex-linked or X-linked. These are the sort of things that on the MCAT tell me future question. So from paragraph 1, I immediately think there's going to be some sort of future question about a mutation, number 1, and maybe about pedigree or genetics or allele distributions in general, since I had that kind of vague autosomal recessive clue in there. So right after paragraph 1, we've already predicted 2 likely questions the MCAT is going to bring in. Let's go a little bit further. Paragraph 2, we could probably outline something like this, right? Vitamin deficiencies, cysteine pathway, blah, blah, blah. But what within this is really likely to be tested? There's some cofactors, some new information, Maribel, definitely James. Right, there's the non-inherited forms. So there's new ones that somehow spring in. Okay? Now there's actually one more piece here. What's the one more new piece? I'll give you a hint. It's a process. Often on the MCAT, if we see process information, that tells us a future question based off of whether or not we understood what that process would do or how it would change the information. You got it, Suhag, absolutely. We had this homocysteine and methionine interconversion stuff. You got it, James, absolutely, William. So I needed to have these vitamins, right? This is because B6 is necessary for this sort of thing. In addition, B12 is necessary for these sort of conversions. Okay, so what's testable is that we need to have these B vitamins in order to have these interconversions. That's a process. Well, what happens if we don't have B6 or B12? This is what the MCAT could ask you. What if we're lacking one? Well, then we can't do that, right? Exactly right, Robert, right, William? What if we had an overabundance? What if we had a superabundance of B6s and B12s? Well, got a lot of extra cofactors. That sounds like plausibly we're helping the enzyme, but it would still be the enzyme. Exactly right, James. It would make the difference. Maybe we could have the process occur in excess, but the enzyme is still going to be that Vmax that stops no matter how many substrate or cofactors we have available to us. But check it out. We had two questions that we sort of spitballed from paragraph 1, and we have at least one new question from paragraph 2. Something about this process, a stop to the process, an extra piece of the process, etc. Now, the chart here gives us a little bit of information. It clearly is our concentration versus our rate, but as soon as you see a concentration versus a rate chart, one concept should immediately spring to mind. At least one term, because the MCAT consistently tests it. Enzyme kinematics is exactly the concept, and Km, Robert, or Vmax. Those are the two most tested concepts within enzyme kinematics, either the Km value or the Vmax top most possible speed. Done. So as soon as I see this, I immediately, even by accident, think, oh, 3 is about the Vmax, and our K is, somewhere 0.5, etc. Immediately. That's the sort of thing that we would pick out. Now, is it worth your time to spend a while analyzing it, looking for other trends? No, because there's really only those two likely questions, and we can read it easily off of the graph. So I'm going to tally it up. Now I have 4 questions potentially predicted. Finally, I've got some sort of process here, the methionine to homocysteine, and it's the pathway. Anytime, though, you see a pathway, think, what happens if we had a problem with transferase? What happens if CBS had been cut out? What happens if we had methionine synthase that ended up being in superabundance? Right? These are the future questions anytime you see a process. All right, I'm up to 5 questions now. Predicted future questions. Finally, we do have a bunch of information here. I could overview it as sort of a mechanism for demyelination and atrophy. There's a big ol' however right in the middle, though, where it's telling me there is also some other stuff. There's also atrophy of the neural tissue. It might have more from this than from demyelination. There's some other question marks happening in here. Anytime you see information that's asking you about a correlation, and somewhere in that paragraph it says more, or while, or also, it's giving you a connection or a comparison between a system that we think might be the most important system, immediately think future question. Anytime the MCAT gives you a comparison or a thesis for some sort of connection or system, think, all right, well, they got to ask me more about this. They're going to probe that information to try and determine, do I know what would happen if we change that system? What if this is true, that there actually is more cognitive defect from atrophy than from demyelination? Does that help or hinder the original thesis about mental retardation? Now, again, I'm not going to force you to answer that right now. That's a future MCAT question we could see on this passage. So right now we've predicted 6 questions. 2 from the first paragraph, 1 from the next one, 1 from the figure, and another one from, again, this last paragraph, and 1 from the overarching system or process they gave us there as a quick visual. That's 6 questions. Let me ask you something. Going into this passage set, knowing 6 extremely likely questions that are about to come up, how do you feel compared to when you saw this, when you took it for your diagnostic? Feels pretty good, right? At least feels better. Even if you're not 100% sure on your membrane traffic or on your myelination, all that, at least you know what you're about to see. You can better predict it. You can better jump in and attack it. Absolutely, James. Less afraid. I love that. I love that. Now, again, the point here, as Suhag pointed out perfectly, is now that we know, we have a better prediction, We're better prepared. We've already pre-thought, gotten our minds in the game for what we're going to have to be answering. So then we jump in with questions like this. All right, what would we see in excess? Okay, well, let's see, in excess. So in excess, if they have homocystinuria, it means they can't do that CBS in the process. If they can't do CBS in the process, we knew this was going to be a question. We predicted it. So we can't make cysteine. So if you see cysteine in any of these answers, get rid of that. Anyone who's memorized some of these already see cysteine? It is answer choice C. Jessica, Catherine, nicely done. Now, we look upstream. I see homocysteine and I see methionine. Now, one of these is in the answers. If you've already done some memorization, you know that A is methionine. Nicely done. Catherine, William, Robert, nicely done. A is methionine. Bonus question, just for funsies. What's B? B is glycine. Maribel, fast. Love it. Catherine, James. Again, to be clear, you do have to have these memorized by testing. So if you haven't already put them on flashcards, absolutely, as Dom mentioned in the last session, put these on flashcards immediately. Get these memorized and out of the way. How about the last one here, D? Nicely done. D absolutely is arginine. And just to be clear, put them up here next to each one of these in case you want to take some quick notes. But D is arginine. Absolutely right. Cool. James, Robert, Catherine, love it. Again, we do have to memorize these, and the MCAT expects us to go a little bit further than just structures. The MCAT expects us to know which ones are hydrophobic versus hydrophilic, which ones have a relatively acidic side chain versus basic side chain. And this is stuff that you can be practicing and putting on flashcards, for the next several weeks until you get them. Don't feel like you're behind the boat if you don't have these memorized yet. You've got plenty of time. Plenty of time. And exactly right, Dom. A good way to get these down. I like to add that we should, when we're drawing these out, if possible, the forms you see in front of you are the ones the MCAT would use if they ever showed you these again. So if possible, something about that form, if you can make a connection to something that makes sense about it. Like, for example, methionine, I think, starts a lot of processes and systems, and it has an S up here at the top all by itself, right, kind of stuck off the end. So I remember it as S. Or things like, again, there are lots of other examples I could bring up. Cysteine, I think of disulfide bonds for cysteine. I think that SH is usually part of disulfide bonds. Again, any way that you can try and memorize these with something that keys you in to, oh, hey, this functional group is clearly different. Glycine is one of the simplest possible amino acids. Well, the simplest functional group that could be attached would be a hydrogen. Again, pulling in all of these different memorization techniques is a great way to make sure you do have these memorized. And thanks, Dom, for that extra little piece. And just to be clear, the right answer was absolutely A. That is methionine, and that's what we predicted. We knew we were going to see this question. Let's do it again. Question number 3. Now, we did predict that we would get some information about gene mutations. Remember? All right, well, here's that question. We knew it was going to come up. So let's think this through. We've got a mutation, and the passage told us that it's reducing function, but it still has some amount of function. So before you match to the right answer, because I bet a few people are chomping at the bit, hold on, is there any answer choice we could get rid of since we know it reduces function and it still works somewhat? Let's get rid of D. It's certainly not silent if it's reducing function. Thank you, Jessica, Maribel, Rachel. Nicely done, Robert. And let's also probably get rid of one more. I saw Catherine said nonsense right off the bat. Absolutely right. Now, nonsense, just to be clear, nonsense is a stop codon early in the process. Generally, this is a completely deleterious mutation. It stops wherever we were in the coding of the protein. It is not likely to have anywhere near the original functionality or enzyme function of the first thing we had. It's completely changed, maybe from 1,000 nucleotides to 100 nucleotides, from 150 to 20. We don't know. Now, nonsense and silent are specific types of mutation. Bonus question. Silent mutation usually affects which nucleotide in the codon? Third codon, aka What is third nucleotide swapping called? It's called wobble. Emily, I love this class. You guys are doing rock star. So silent mutation, wobble, third nucleotide change, and definitely not what we're seeing here, because it would be doing the same thing. Similarly with nonsense, it's cut. Now, at this point, even if you weren't sure between missense and frame shift, there's an issue. And that's that a frame shift mutation is a larger category. Nonsense, missense, and silent are called point mutations. Frame shift is a brand new subcategory that could end up giving you nonsense or missense or potentially silent. So frame shift is not a mutation, it's a category of mutations. So at this point, a critical thinker would jump in and say, gotta be B. It is missense, because frame shift is not specific enough to tell us which of the other ones we would end up with. Similarly, as Maribel is pointing out down here, missense is either usually an insertion of one or two nucleotides or a deletion of one or two nucleotides, which would change the actual frame of reading for the entire rest of the protein. Now, to be clear, the other reason that frame shift is wrong is because it doesn't tell us what happens. For example, it could be frame shift in the first couple codons, and then that would just destroy the protein. Or it could be a frame shift in the last one or two codons, right? And if it was only a frame shift in the last one or two codons, then it could still work. I've got a bonus question for you. You know I'm all about bonus questions at this point. If frame shift could work if it's the last one or two codons, or it would be totally wrong if it was anywhere in the middle of the chain, does that mean that an answer that's maybe true could like sometimes be right on the MCAT? Because like maybe true, like I could find one or two ways to make it true. Never, ever. Anytime you see an answer where you stop and you vacillate and you say, well, you know what, it's asking what color the sky is, and the sky for a little bit of the nighttime is like dark blue, and then at sunset it's sort of like red, so I'm thinking like maybe red could sometimes be right. No, the right answer is blue. The sky is blue. It is always blue. Even if you can think of one tiny exception in biology that somehow happens, that's not what the MCAT's getting at, and you know better. So this is going to take a little bit of time to get used to, but the MCAT is not your advanced genetics class. Your MCAT is not the biochem class. Your MCAT is a holistic survey course. It wants to know, did you get the gist of all of the foundation content you were supposed to do in the first couple years of undergrad? And if you can get that, we grab the point, we move on. We don't try and get trapped by any of those other convincingly tempting wrong answers. Okay, now one more thing. We expected to see something about this chart, and they're absolutely giving us information about this chart, but there's a key piece to the question. Now, I've been training you. You know what to pay attention to. What's important in question number 4, that if you were speed reading, you might have missed. The not, which could not have been obtained. Emily, Ariel, James, nicely done. So we're looking for the one that would not be likely. Now, we need to know two things. We need to know what Km is, and we need to know what Vmax is. Absolutely. So as far as the concept here, we're really trying to tackle what would be affected or what would be changed, or which of these could not fall within the curve, because 3 of these will have been somehow possible, and one of them has to be impossible. So what we can do is pull back up, if we don't remember this, information about what Km is and what Vmax is. So I'll tuck this away on the right-hand side, just to be clear. But the idea is we want one that's not possible. So for a particular Km value, is there a Vmax that's impossible? Or the flip side of that, for a particular Vmax that's possible, does it require the Km to be too big or too small for our chart's information? So we'll get there, Jessica. Let's look at figure 1 first. Now remember, Km is that value that we pick, 0.1, in this case would fit within our chart. 6 would not necessarily fit within our chart. So here's my question to you. If we don't see 6 on our chart, can we just like say probably the chart keeps going? Probably it's all good, you know, it's like move over like 2 more? No, we can't actually, right? We absolutely cannot make that connection. So then D, 6 with a Vmax of 1, well, based off of an interpretation of what the chart looks like, that would be well within the Vmax line. That would potentially be great. Similarly, A, 6 and 30 sounds really way too big, but I just don't know what happens on that curve later on. I assume that it's going to be exactly the curve I'm seeing, but I just can't see it. So I'll hold that for a second, because maybe on test you would hold it and be concerned. But let's look at the other 2 options, 0.1 and 1. Well, somewhere down here around 0.5 or 0.1 or so, Km hits a Vmax of a little over 1, or almost 2. So 0.1 Vmax of 1, answer choice B, is absolutely within the values that we would expect, right about here. So if that's within the values we would expect, it's not the outlier. So then it's got to be answer choice C. 0.130, way up here in outer space, is absolutely not something that we could hit, considering our Vmax is somewhere around 3 or 4. Now I know what you're thinking. You're thinking, but hold on, Eli, wouldn't 6 and 30 also basically be impossible because that's way above this line? What did I say? What did I say about 6s in general? Now this is, again, not just for this chart, but for any time we see this type of question. Yeah, they're not on the chart. And going back to what we talked about in the last question, if it's maybe true and maybe not true, it's instantly a wrong answer. Done. If I have to sit there and think to myself, gosh, maybe, I guess, if the curve was, then no. Cross it out, move on. It has to be answer choice C. Now at this point, we do have another passage lined up for you to go down through. The theme essentially is incorporating experimental passage information, how to read it actively, how to do questions actively. And as far as the tackling of this, we can do this. But my goal of spending a little bit of extra time walking down through the last passage and really trying to make sure we understood what to look for was so that you could do this. We could use this passage, right? This happens to be a passage from the Diagnostic, right? Passage 5. We could go down through the HIV passage and talk about what's most important, what type of content is likely to be tested. But my goal was to turn you loose and have you do that process. If passage 5 was the hardest one for you, go for it. If passage 2 is the hardest one for you, do that one. But since you already took the test, you can do it even more efficiently than other students. So what I want you to do when you're going through and doing this on your own is essentially what I was walking you through. What could be tested from this paragraph? What could be tested from that paragraph? But since you already know what the answers are and what the questions are, you can go one step further. Maybe here's what I had for my roadmap, but instead of just asking yourself what could be tested, ask yourself what was actually tested. They actually did test that TAT protein consistently throughout the questions from this passage. And they talked about activation of T cells and how to make that connection. So we can go back and we can say, hey, paragraph 1 actually had a lot of information that came from the question set. Paragraph 2 about TNF, yeah, it's something about activation, something about an experiment that was designed. We needed paragraph 2. That was at least one question. See what I'm getting at? What you're trying to do is what the best test takers do. Weed out the chaff or the fluff from the paragraph and pull out only the grain of information that's actually important to get the questions. Because, bonus question, what partial credit does the MCAT give you for reading the passage? 0, right? There's no oral review at the end. There's no book report summary that you write on your scratch sheet and they give you partial credit for. Nothing. So the point of reading the passage is to read it basically like a murder mystery. Like you're looking for clues. And if you can pick out this clue will likely be tested, then you're doing what the best test takers do. Now again, your outline is to try and keep track of everything. So notice how this outline for paragraph 2 immediately hints to us we're going to see a question. Something about maybe targeting. I bet we're going to get a question about that. Activation, great. And then what was required? Perfect. That's at least one question. Again, same thing with talking about the details within that paragraph. As soon as you see these words, like believed to be, maybe, or a comparison, immediately think we're going to have that question, delving a little bit deeper into that to figure out what's actually important. Something like this, a big list, what do you think most students are going to do when they see a list like this on test day? I know we're close to the end of our time together, but I really want to make sure we talk about this because this is the sort of thing you need in order to review the rest of your diagnostic. And I said most students, Emily. We're better than most students at this point. But what most students do, they would freak out, Ariel. They might completely ignore it, Rachel. Or, you got it, Jessica, waste a minute and a half, reading the whole thing in detail, trying to figure out what each one of those different experiments would give you as a result. Oh my goodness. We don't even know whether they're going to ask you about C1 or C2 or D2 or B yet. Until we know what's going to actually be tested, doing that sort of long, drawn-out comparison is getting in the way of answering questions. Now, the ideal test taker would scan through the list and notice if there was something that was an outlier. Like A, cells alone, we don't expect to really see a whole lot of difference. Or D1 and D2 dealing with slightly different amounts of PHA with TAT peptide. Okay, comparison in there. So on my roadmap or my scratch paper or my outline or my cheat sheet that I'm keeping off to the side, I'm going to jot down D1, D2, something about TAT peptide difference. C1, C2, again, TAT peptide difference with no PHA, right? Quick little note, so when the actual question comes up that says PHA on my plate with TAT peptide in this amount, I know exactly where to go back to in order to compare that type of experiment. Now at this point, we are pretty much right here at time, and my goal for your sessions is to give you every single second valuable information, tests like critical thinking, and a way for you to go back and effectively do your diagnostic review. So without further ado, I'm going to let you go through the rest of this for practice, and certainly go through the rest of your diagnostic, doing the exact same sort of thing. Evaluation, review, how could you read more effectively next time, how could you eliminate more efficiently next time.